See now, we'll go in detail about the structure of microsporangium and formation of microspores. We'll go in detail. Already you wrote a diagram. Okay, you draw a diagram with the help of that diagram. I will explain. I will explain that with the help of a rough diagram. Usually, if you see anther wall, there are 4 layers. 4 wall layers are present in anther wall. Okay, what is that? The outermost layer is called epidermis. This outermost layer of anther wall is epidermis. That epidermis is made up of single layer. Epidermis is thin outermost layer. There is a thin cells are present and it is outermost layer, and it is responsible for protection and dehiscence. What is dehiscence? Dehiscence means once anther matures and there is formation of pollen grains, that anther will burst out. That is called as dehiscence. Bursting out and release of pollen grains into atmosphere or environment is called as dehiscence. Dehiscence is carried by epidermis. Below the epidermis, there is presence of another single layer cells called endothelium. Endothelium. Okay. Second layer is endothelium. This endothelium is thick layer. Okay. This is also single layer only. Epidermis is thin outermost layer and it is single layer. Single layer of cells are present in epidermis and endothelium. Endothelium is also responsible for dehiscence of anther. Anther dehiscence is mediated by both epidermis and endothelium. Then third one, below the endothelium there is presence of 1 to 5 layer wall called middle layers. Third layer is middle layers. Third. Usually middle layers are 1 to 5 layers of cells are present, and the function of middle layers is consumed by developing microspores. When microspores are developing, that developing microspores consume that middle layers as they. Okay, that means it will undergo degeneration when microspores are developing. Then innermost layer present in anther wall is tapetum. Tapetum is innermost layer, and this is also single layer only. Single layer. It is a single layer. Cells with dense cytoplasm and multi-nucleate condition. So these are middle layers. Below that there is presence of another layer called tapetum. This tapetum is a layer, innermost layer, single layer only. Dense cytoplasm is present. What is dense cytoplasm? Cytoplasm means inside the plasma membrane, number of organelles are more, and cytoplasm has more chemical substances. Thick cytoplasm, that is dense cytoplasm with multinuclear condition. Okay, more than one nuclei. That means two or more nuclei are present. That is tapetum. Okay, tapetum function is nourishment. I will tell you again. I will write from there. Okay, this is tapetum. So below that, inside that tapetum, there is the presence of a tissue called sporogenous tissue. This sporogenous tissue is responsible for sporogenesis or formation of spores. I will erase this and start with this. Tapetum. This will continue this one. Tapetum. Okay, tapetum is innermost layer, single layer, then cytoplasm with multinuclear condition. It is responsible for nourishment of developing pollen. When microspores are developing, to that developing pollen grains, tapetum gives food material. That's why it is called as a nourishing tissue. It is responsible for the nourishment of the developing pollen. Okay, or it is a nutritive layer. Okay, and sporogenous tissue is there. That is not a part of anther wall, that is present inside the anther. And tapetum is there now. Tapetum is especially there is a tapetum called as secretory tapetum. That secretory tapetum secretes two types of substances. One is Calase enzyme. Calase enzyme. Calase enzyme. What is Calase enzyme? This enzyme is responsible for callose in pollen tetrad. Callose enzyme. Callose is a substance which is present in pollen tetrad that is broken down by callase enzyme. See, what is the callose? How it is broken down? What is its function? You see the significance of that breakdown of callose by callase. Usually, by meiosis, what happens? There is a cell. That cell undergoes meiosis, first reduction or division or integration or division, and there is formation of 4 haploid cells by meiosis takes place in reproductively active cells. In reproductive cells, it is taking place. There is formation of 4 haploid cells by meiosis as a tetrad. This is a tetrad. Okay, the cells in tetrad are connected each other with a substance called as callose. Callose is a chemical substance present between spores in a tetrad. Okay, that on that there is action of enzyme callase. This is callase. This is callose. Callase is an enzyme that breaks this substance and it is responsible for the release of individual spores from the tetrad. Individual pollen are released from the tetrad by the breaking of callose present here with an enzyme callase. Okay, that is the function. Okay, then another function is there, another secretion. It secretes what tapetum secretes another structure called Ubish bodies. Ubish bodies. What are the Ubish bodies means? Ubish bodies will get covered with sporopollen, and this will increase thickness of pollen wall, pollen grain wall of the pollen grain. What is that sporopollenin? Sporopollenin is very, very important substance. That is a highly resistant or most resistant chemical substance is sporopollenin. This sporopollenin is present on surface of pollen grain. In pollen grain structure, there we will see as epidermis, endothelium layers are there. Like that, pollen grain has a different layers like exine and type. Exine and type. There we can observe this presence of sporopollenin. Sporopollenin is the most chemical substance present here. Okay, what is that means sporopollenin is most resistant chemical substance. With the help of sporopollenin as a most resistant chemical substance, only pollen grain has most time of viable period. That means pollen grain can be active for very long times, even thousands of years also they can survive due to presence of sporopollenin. Okay, here this apertin is secreting a structures called as Ubish bodies. That Ubish body is covered with sporopollenin. Okay, Ubish bodies are secreted, they are covered with sporopollenin. That is formed on the surface of the pollen grain as outermost layer on pollen grain. Outermost layer of pollen grain is formed with the help of Ubish bodies and sporopollenin. That will increase the thickness of pollen ball and gives resistance to the total pollen grain. Okay, this is the function Ubish bodies. Okay, and apertin is responsible for formation of another structure. Another structure is formed by apertin known as pollen kit. What is pollen kit means? Pollen kit is an oily, thick, and sticky substance on pollen grain. Pollen kit is an oily, thick, and sticky substance which is present on pollen grain, which is made of oils, lipids, and carotenoids. That is made with lipids and carotenoids. That is called as a pollen kit. What is the help of this pollen kit means? It is responsible for attachment of pollen to insects in entomophily. Entomophily, entomophily is a type of zoophily. What is entomophily? Entomophily is insect-mediated pollination. What happens in insect-mediated pollination? In insect-mediated pollination, first insects will land on a flower where male organs are present. When it is landing on a flower, if that flower has male organs and there is a production of that pollen grain, that pollen grains will attach to the surface of the body of insect. Then the same insect will go to next flower, another flower. The pollen grains are now carried to second flower. That is insect-mediated pollination. In that flowers, in that kind of flowers, especially entomophilous flowers, pollen grains are covered with pollen kit. That pollen kit is oily, thick, and sticky substance. As this pollen kit is present on surface of pollen grain, and this is oily and sticky, that will attach to the insect very well. Okay, that will be attached to the insect, so that pollen grain will easily attach to the insect body. That will be easily carried for pollination. That is one advantage. Okay, right. This is about the structure of microsporangium and anther wall layers. Okay, now then I will show the matured microsporangium with screen sharing. After that, I will explain the formation of microspores. Okay, see now. See now, this is a matured sporangium, microsporangium with pollen formation. See here, I told you there are 4 wall layers. What are that 4 wall layers? Epidermis, endothecium. See where epidermis is mentioned. This is the epidermis. This is the epidermis. Epidermis is the outermost layer. Then endothecium is mentioned. Where that endothecium is mentioned? Endothecium is next to epidermis. This is endothecium here. Below that epidermis, there is endothecium. And middle layers are there. Middle layers are those which are present next to endothecium. These are middle layers. And inside that there is a presence of tapetum. Tapetum is the nutrient tissue. This is a total tapetum with multinucleate condition, dense cytoplasm. This is a totally tapetum. Okay. Then inside that there is presence of what? Sporogenous tissue. This is the sporogenous tissue. In this sporogenous tissue, there is a formation of microspores with the help of meiosis. This is a sporogenous tissue. Okay. Sporogenous tissue where meiosis is taking place. See, this is connective. Connective is the tissue that is responsible for attachment of 4 sporangia. Okay, this is connective. And I told you this is the matured sporangia or anther that shows matured sporangia with matured pollen grains. Pollen grains are also present here. See here, these are the sporogenous tissue are there, cells are there. Now these are the cells that will develop into pollen grain, and that pollen grain are produced in large number. Once this will become fully matured, then this sporangium and this sporangium both will unite. They will combine and they will dehisce. They undergo dehiscence and release of the pollen grain. Okay, I will share another diagram. You can check. See this one. This is the matured anther and dehisced anther, where dehiscence also taken place. That means the dispersal of stamens, or dispersal of pollen grains also took place. Here also we can observe that where epidermis is here, epidermis is here, endothecium is this one, middle layers are these, but here tapetum is absent. See here, these are middle layers, right? Tapetum is absent. Why tapetum is absent? I told you tapetum is a nutritive tissue for developing pollen grain. As the pollen grains are developed, tapetum is fully consumed for the development of pollen grain. After the development, what happened? Actually, there should be 2 microsporangia on each lobe. Okay, 2 lobes are showing here. What are the 2 lobes? This is totally 1 lobe, and
This is totally one lobe. This lobe has one microsporangia, that lobe has one microsporangia now. But actually two microsporangia, here one microsporangia, here one microsporangia. Two microsporangia should be there. But after maturity, two microsporangia became united. See now. Two microsporangia are united. This will take place with the help of connective only. Both are united and this is the place of dehiscence. See, what are the parts of tissue of anther wall? They are responsible for dehiscence. Epidermis and endothelium. This epidermis and endothelium will break and dehiscence takes place. See, so that pollen grains are releasing outside. See, right side, pollen grains are releasing now. That pollen grains are coming out with the help of the dehiscent part. This is the dehiscence part. Okay, this is a matured anther and dehisced anther. Okay, now I will go to board again.
Right. See now, how that microsporangium is responsible for formation of microspores. Microspore formation. What is microspore? Microspore is nothing but pollen grain. See, microsporogenesis. Microsporogenesis means formation of microspores. Formation of microspores. The microspores are also called pollen grain. So formation of pollen grain or microspore is called as microsporogenesis. How microsporogenesis takes place means usually inside the pollen sac. What is pollen sac? Inside the tapetum, totally it is called as pollen sac. Inside that pollen sac, that means in the pollen sac or microsporangium, there is presence of sporogenous tissue. Microsporogenous tissue is there, or microsporogenous cells are present. These microsporogenous cells first undergo mitosis. Upon mitosis, there is formation of PMC, pollen mother cell, or microspore mother cell. Microspore mother cell or pollen mother cell are formed from the sporogenous tissue which is present inside the tapetum. Then this PMC, pollen mother cell or microspore mother cell, undergoes meiosis. By meiosis, there is formation of microspores or pollen. These are formed in the form of tetrad. 4 haploid microspores are formed in tetrad. That tetrad is formed. That tetrad may be isobilateral, or tetrahedral, or linear or T-shaped. What are that? Isobilateral means there may be presence of formation of tetrad with equal number of cells on lower and upper surface. That means two spores are formed on lower surface and upper surface. That means cell on another cell like that, 4 cells are formed. Two cells are here. On each cell there is another cell. That is isobilateral. Tetrahedral means four cells are formed side by side. 1, 2, 3, 4. Here 1, 4. But side by side, not one on another. Linear form means which are arranged in line. T-shaped means like this. Four haploid spores are formed. That four haploid spores are formed in a tetrad. That tetrad can be like isobilateral or tetrahedral or linear or T-shaped structure is formed. This is normal. Okay, but some special cases we can observe. What are the special cases means? Usually, four haploid microspores are formed from one meiotic division. One meiotic division takes place. Upon one meiotic division, there is a formation of four haploid microspores. But there is formation of only one microspore. Only one microspore formed on meiosis. That means only one pollen grain. Usually there is formation of four pollen grains, but in some members like Cyperaceae family, in some plants like Cyperaceae, there is formation of only one microspore on meiosis. That is one important feature. Then, if you see another one, in some plants, there is a formation of pollen grains as a pollinium. What is pollinium? Pollinium is a bag or sac-like structure of pollen grain. Bag or sac-like pollen. The pollen grains are developed in bag or sac form. They are said to be pollinium. This pollinium we can see in some members like Asclepiadaceae or Chilidaceae. Asclepiadaceae members and Orchidaceae. Asclepiadaceae, best example, Datura. What is that pollinium means? Usually pollen grains will be in the form of tetrad, but here a pollinium is formed. Like here, a sac-like structure is formed. Inside this sac, there is presence of number of pollen grains. Number of pollen grains are present inside this bag. This type of pollen grains group is called as pollinium. Pollinium is another one. Okay, another condition we can say polyspory. What is polyspory means? More than 4 pollen are formed. Usually 4 pollen are formed, that is a tetrad is formed. Okay, more than 4 spores are formed. That is called as polyspory. This polyspory we can observe in Cuspida plants. Cuspida. Another condition is there which is called as compound pollen. What is compound pollen means? Compound pollen is a condition where tetrad is there now 4 haploid spores are formed. Tetrad do not separate. As the tetrad is not separating, 4 pollen grains will be as a group. That is called as compound pollen. This compound pollen we can observe in a plant called Drosera. Drosera is best example. These are some other cases of microsporogenesis. Usually, microsporogenesis takes place in plants with the help of microsporogenesis. Okay, the sporogenous tissue which is present inside the pollen sac will undergo first mitosis, then meiosis. Upon mitosis, there is formation of pollen model cell. Then they will undergo meiosis and produce more number of or large number of pollen grains as a tetrads. That is normal. But there is no formation of tetrad, only one pollen grain may form inside the seed. And the pollen grains may be formed as a bag-like or sac-like structure in Asclepiadaceae and Orchidaceae members. Okay, more than four pollen grains may be formed as tetrad. Tetrad has usually four only, but there may be more than four. That is found in Castuta. And usually after the formation of pollen tetrad, callos will break down by callase and individual pollen grains will get separate. That is normal. But in some plants, the tetrad may not separate. That remain as a compound pollen as in Drosera. Microsporogenesis. Just to complete that. Okay. Right? With that, microsporogenesis is complete. Today class is also over. In next class, we will go to the structure of pollen grain, then how there is formation of male plant, and then we will go for female reproductive organ like the structure of carpel and the remaining properties of carpel and how there is formation of megaspores and megagametophyte that we will see in next class. If you have any doubts, just ask me now.